Oh, there I see the candidate. Mr. Cai Zhang, welcome. Can you understand me? Can you understand me? Can you hear me? Can you see me? Would you please unmute your microphone so that we can hear you?
Yeah, now it's okay.
Okay. And if you like, would you please adjust your camera a little bit because we don't see your upper part of your head. So if you can move it a little bit downward, a little bit.
Sorry, I can't change my camera. Yeah. This is okay?
This is fine. This is fine. Yes.
Okay.
And can you hear me clearly?
Yes.
Yes. So I will start in about 2 minutes, and I will start the ceremony with introducing you and the supervisors, and then I will invite you to start with a presentation of the summary of your thesis. So when I invite you, you can start with your presentation. And please don't forget to share the slides with us so that we can enjoy your presentation.
Okay.
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 If you do not understand the question, so you can ask to repeat the question if you do not understand. And we try not to speak too quickly.
Okay, thank you.
And after that, we will have 45 minutes of opposition. I hope that you are not too nervous. And are you prepared for starting the ceremony? Do you have any other questions?
Okay, no problem.
Okay. Thank you very much. Then I would like to ask our ICT supporter to start the live stream.
So now I can share my screen.
Hereby, I open this academic ceremony at Maastricht University. My name is Ton de Groei. I am the pro-rector and the chair of the degree committee. And we have now in this ceremony Mr. Cai Zhang, and he will defend his thesis in public. And his thesis is entitled Immunotherapy, a potential treatment strategy for depression. And I will like to welcome all the members of the degree committee, and in particular, the 4 supervisors. And the supervisors are Professor Harry Steinbusch. He is Professor of Cellular and Translational Neurosciences at Maastricht University. Professor Thierry Charlier, he is professor at the Institut de Recherche en Santé, Université de Rennes in France. Professor Cai Zhang, she is director of the Institute for Marine and Nutrition at the Guangdong Ocean University in China. And the 4th supervisor is Dr. Pawlowski, and she is professor at the Institut de Recherche en Santé, Université de Rennes en France. I will introduce the 6 opponents later during the opposition. And I welcome, of course, the audience of the live stream, and particularly those viewers and followers in China. Mr. Cai Zhang, may I invite you to give a summary of your thesis, and I wish you success in the coming hour. The floor is yours.
So now I can start. Okay. Can you see? Okay. Dear Director, Coroller, family and friends, I'm glad that you can attend my PhD defense. In the next 15 minutes, I will summarize my thesis. My topic is immunotherapy, a potential treatment strategy for depression. First, the depression is a mental disease. The main symptoms is this, including anhedonia, hopelessness, anxieties, and the depression is mainly caused by the non-adverse life events, stress, inflammation, and autoimmune disease, hormonal disorders. So in the last past decades, there are some classical hypotheses proposed, such as HPA axis, neurotransmitters, inflammation, neuroplasticity. But based on this hypothesis, the antidepressant is not very good and with serious side effects. In the recently, there are many studies have found that there are many immune biomarkers abnormalities have been found in the depressions. So this is why our study focuses on the immune systems. And so how the stress induce the immune systems and the brain. Here we can see there are 3 different ways. The first is the stress activates HPA, and then over cortisols, over release the cortisols. And the second is stress activates the vagus and sympathetic chains. And the third is stress induced the molecular release, such as the neurotransmitters, and then they will act the immune cells through some receptors on the immune cells. So when stress activates the immune systems, and then this will induce the inflammatory cascades through these inflammatory pathways. Then these cytokines, protein, and other factors will through the blood-brain barrier, and then communicate with the central cells like the glia and the neurons. But how this substance through the BBB? First, there will these small molecules will through these Nick junctions, and the second, some cytokines will transferred through by the receptors on the blood vessels. And the third is the activity immune cells, such as lymphocytes, macrophages, can pass the BB directly. So in the last decades, there are many studies have focused on the macrophage, like Professor Nemmer and so on have did many works to demonstrate the macrophage theory of the depressions. But recently, the central lymphocytic vessels have been found. But how the central lymphocytes changes and regulates the neuroinflammations is not clear. So that's why our study focuses on the lymphocytes. First, the lymphocytes can be divided into like the CD8 cytotoxic T cells and the hyper CD4 T cells. In here, the CD4 is popular studied because it can differentiate into different subtypes, such as the Th1, Th2, T17, and T regular. And then these subtype cells can release different cytokines like these interleukins and then work. So how this cell and cytokines involved in the depressions? Generally, the depressions, in the depressions, we find that the decrease CD4 Th2 regular while increase CD8 pro-inflammatory Th1, T17, and their cytokines. See, this slide is detailed to describe the stress induce the potential changes in these lymphocytes. First, the lymphocytes are born in the bone marrow, and then they will mature and differentiate into different subtypes in the thymus. Then they will work in the lymphatic tissues like the spleen, lymph nodes, or the blood. And then when give the stress, these cells, some cells will recruited into the central through the lymph vessels. And then they will through the BBB to communicate with the glia and the neurons, and then induce, but how they induce the neural inflammation and the neurotransmitter is not clear. So our study proposed some questions. The first is how the peripheral and the central lymphocyte changes in a stress-induced depression. The second is these lymphocytes, how to communicate with microglia, and then regulate the neural inflammation. So for answer these questions, we use some immunomodulators to treat. But if these modulators, immunomodulators effect or how they regulate the lymphocytes and the neural inflammation is not clear. So let's see, here is some the immunomodulators. First, we use the thymus alpha-1, is T-alpha-1. Why we use this? Because it can enhance the immune response and also regulates. So we want to see if the T-alpha-1 can have the antidepressant effect. We find it can activate the MAPK pathway and regulate apoptosis signals. So we also use the ERK and BCL inhibitor to further demonstrate the T-alpha-1's functions. The second, we use these three drugs to target the Th1 lymphocytes, M1 microglia, and MAPK inflammatory pathways to study how lymphocytes and microglia can regulate the neural inflammation. And also we compare their antidepressant effect. So here is the experiment protocols. First is the stress models, and then the drug treatments, and then behavior evaluations. And then we will connect the samples and analyze this. So here are some results. The first, because the males are popularly used in the neuroscience, but in the clinic, the women are about twice as bad as men with depression. So we designed an experiment to study and compare the male and the female in the stress-induced depressions. And we find some of the difference, 6 difference in the behaviors, lymphocytes, hormones, inflammation, and neurotransmitters, especially in the lymphocytes. They both have 6 difference before and after stress. So in the part 2, we find the stress activities M1 microglia and Th1 and lymphocytes, and then they over release the pro-inflammatory cytokines. And then these pro-inflammatory cytokines will induce the dysfunction in the neuroplasticity, neurotransmitters, and thereby induce the depression-like behaviors. So here we use the mentions about the immunomodulators to interventions and find they are all improve the depressive symptoms. And also we find that target Th1 is the best choice. And these results will have been introduced details in the chapter 2 to 4. So in the summaries, we have find that. The first is this study first found the 6 difference in the lymphocytes before and after stress. This reminds the 6 difference should be considered in the depressions. The second is, the second is our study targeted Th1 lymphocytes and find it have a good antidepressant effect. And also we compared with the microglia inhibitors. And this reminds the lymphocytes and the microglia may be interactor to regulate the neuroinflammation and then induce the depression. So, and also we find that target Th1 was a better choice. And the third is T-alpha-1 was first used to treat depression in these studies. And we also demonstrated that T-alpha-1 had a good antidepressant effect through ERK pathway and BCL singulars. So for future studies, we have some study nights in like the genders or 6 difference, because we have found the 6 difference in the lymphocytes. So if we give the immunotherapy to treat, if there are some difference, or if in the clinical is have some difference. And the second is because we just do the immunotherapy in the animals, but we don't clear if it affects in the clinical. Yes, that's all. Thank you. Thanks.
Yes. Thank you very much, Mr. Zhang. I will wait until your presentation will be finished. Yes. Thank you very much again, Mr. Zhang, for your clear presentation. The opposition will now be opened by Professor Martinez. Professor Martinez is a professor of neuroinflammation of neuropsychiatric disorders at Maastricht University. Professor Martinez, the floor is yours.
Thank you, dear Pro-Rector. I want to give my compliments to the candidate. And I would like to also give my compliments to the promotion team for a nice and very detailed thesis. I would like to ask you some questions now regarding the choice of the animals and of the rodents, for the animals, and also of the behavioral tests. I will start with the question about the induction of depression-like behaviors. You explain in your thesis that in mouse and rats, there are no differences in these tests. So they are both performing well in the same test. Well, I am a little bit puzzled by this because there are differences in the immune system of mouse and rats. And you are trying to demonstrate that you can, the immune system has an important role in depression. So I am wondering, can you elaborate why you think that there is no difference in your behavioral tests between mouse and rats when there are differences in the immune system, for example, regarding the susceptibility to autoimmunity? So when you develop autoimmunity in animals, in rats or mouse, they develop it differently because they have differences in the immune system. For example, rats and mouse do not activate in the complement system in the same way, or they don't give the same antibody responses. So could you elaborate about that?
Thank you for your questions, professors. I would like to, if you want to know, I use the different samples between the mouse or the rat, right? Yeah. Or some also in the model or something, yes, right?
I want to know if the difference between the rats and the mouse, that you do not see a difference, but the immune system is very different. So how can it be? So how can it be that you don't see the behavioral differences when the immune system is different, you know? So that's my question.
It's you mean in the mouse? I just checked in the peripheral the lymphocytes, but not the central, like the meninges. Yes, this is the problem. Actually, I do this experiments first, and then when we have the then we do another experiment for further study in focus on the central, not just focus on the peripherals. Yes, but for the different, I think.
Is
so sorry, you can repeat. Yes.
So maybe the organs that you are analyzing are different in the different rats and mouse, and that's why you see maybe no difference in the response. I don't know, but maybe I can move on to the next question regarding your model of depression. So you use a chronic, unpredictable, mild stress model for depression, but there are many other models that you can use or tests that you can use to study depression, like the force swim test, or like the tail suspension test, or the sucrose preference test. Could you explain to me why you use only these tests, and if it's not necessary perhaps to use more than one test to demonstrate the effects of depression?
Thank you. Your question. You mean why I use these behaviors to demonstrate the depressions, right?
Yes.
Yeah. Because we can see in our the symptoms, the main symptoms of depression. First, is anhedonia, but in the animals we often use the sucrose to demonstrate this. And then when we use the open field and elevated plus, we to demonstrate the anxieties. But here we not demonstrate the hopelessness. This maybe we can use the swimming or for swimming to demonstrate. But this can cause many bigger stress. So we just use the demonstrate the anhedonia and anxieties. Maybe in future we can use this.
Okay. Yeah. Thank you for your answer. So maybe we could move on to another question I have regarding the drug. So you are using minocycline, so mino, and then you inject this in animals intraperitoneally. Now I am wondering, because you inject them for 60 to 80 days intraperitoneally. So and also T-alpha-1 in chapter 3, you also injected in this way. So I mean, I know that intraperitoneal injections are very invasive, especially when done repetitively every day. So are you not afraid that they do affect your stress response in the animals?
You mean why I how I give the drugs, or how the drugs affects, right?
How the method of injection of your drug, that is in this case is intraperitoneally, IP, how the IP injection of the drug can affect the stress levels of the animals, because you need to do this for 60 days every day. If you thought about that, perhaps.
Yes. Sorry. Because the first I will build the stress models, and then in the last 3 weeks, I will give the drugs to treatment.
Okay.
And because the drug, sometimes the drugs can't more than 3 weeks. especially these drugs, not like some nutrition or something that you can use long time, but the drugs basically not long times.
Okay.
And
okay, so and what about the drugs? So when you inject them in the periphery, because you inject them intraperitoneally, do they reach the brain? So is there any evidence of how much of this drug can penetrate to the brain?
Sorry, can you repeat?
You may give a short answer, please.
Sorry.
Yeah, so just to repeat the question very briefly, does the drug, the MINO drug or the T-alpha-1 reach into the brain when you inject them intraperitoneally?
I reject the use the IP, and generally the T-alpha-1 effect maybe after 1 hour they can peak, they can arouse the peak, the drug concentrations. So when I use the inhibitors, after 1 hour, and then I add to the inhibitors. Yes.
Okay, thank you for your answer, and I give my word back to the pro-rector. Thank you.
Sure, I just
thank you, Professor Martinez. The opposition will be continued by Professor De Juc. She is director of the PhD research at INSERM at the University of Rennes in France. Professor De Juc.
Thank you, Pro-Rector. So first, I'd like to congratulate you because I found the subject was really interesting, and so are the results. And there is many questions about the underlying mechanism. So we just go through a few of them because we have limited time. But to start with, I have a general question on the use of immunotherapy in patients for depression. You said one of the issues with the current treatment is the recurrence rate. So it is patients are cured, but for a short time or a limited time, and it's coming back. And I'm wondering, how is it going to work with this immunotherapy? Because as I understand, it is not the primary trigger of depression. It's more like a secondary response or an amplifier. And if the primary trigger is still here, when you stop the immunotherapy, depression might come back. So how do you see that? How sustained do you think this kind of immunotherapy? Can it cure depression, or is just keeping it at bay for a short time or a limited time, and then it will come back because the stress will still be here, or whatever is the primary trigger of this immune inflammation?
Thank you for question, Professor. But I don't understand your questions. Can you just short to focus?
Okay, so as I understand it, and tell me if I'm wrong, it's not the inflammation that is triggering depression as a first cause. It's like there's some stress or other causes that induce depression, and then it triggers an inflammation, and this is what you're going to try to cure with immunotherapy. But whatever is the primary cause, the first cause, might still be here. So how long can you cure people with immunotherapy? Will it, as soon as you stop immunotherapy, depression will come back? What's your thought?
Okay, thank you, Professor. I think you might want to know is the immunotherapy is can keep a long time to treat the depression, right?
Yes.
For these questions, I think the first is about the immune systems, because the immunotherapy is a treat for the immune systems, and so when our immune system likes the lymphocytes changes or something, and then they release the inflammatory environment in the brain, and there will changes, but I think it's not very short. It will keep a long time, but I don't know if it's okay, if it's right, because I never do these studies. Maybe if we can, in the future, we can add some, such as we add a positive antidepressant like the fluoxetine, and then to compare, and then to look if they are okay, and just like the 1 month or 2 months or 1 year, if it's still okay. But this needs some time to do this, so I'm not sure if it can keep a long time. I'm sorry.
Okay. And in the same line, are there any studies showing if there's some epigenetic changes in the, I understand microglia are central in triggering this inflammation, they're activated. So once you got this activation of microglia, does it induce epigenetic changes? That means they'll be more prone, to get activated again and trigger depression. And that's also maybe something that could explain that for some patients, depression keeps coming back. Is it something that you're aware of? It's a naive question because I don't know myself, but are there studies about that, about, epigenetic changes? So as you know, our environment will modulate the expression of our genes. And does it happen in microglia, for instance?
Thank you. And can you just repeat again? I'm sorry.
Okay.
I'm not very good English for this. And can you short? Yes, just a short to say this.
Okay. I have another question about your model. So it's a stress-induced model. And in the same line as what's been asked a bit before, but how representative it is of other models of depression or other triggers of depression. And I was also a bit surprised in your introduction, you don't mention the microbiota, which is a very trendy subject at the moment. And, could what we call a dysbiosis, so a dysfunction, a problem in the microbiota, in the gut microbiota, as a trigger of depression? What do you know about that? And can you talk about how it compares with your stress-induced model? Has it been studied? Does it trigger the same kind of inflammation as what you saw?
Maybe you want to know how the stress can induce the depression, the mechanism or something, right?
I want to know what you know about gut microbiota. You understand gut microbiota? The gut is, we say, is the second brain, and you've got this all these bacteria in the gut that can get disturbed, and for some instance, as far as I'm aware, it can impact on the brain and potentially on depression. So, are you aware of these studies? What do you know about these studies?
I don't guess something, but if maybe sorry about this.
Or other cause of depression.
Maybe you want to know the gut and the brain, and then how to induce the depressions, right?
Yeah. What mechanism? What do we know, or what we don't know about these triggers? Do you know about the another source of depression being something completely independent from stress, or at least not directly stressed on the brain, but something linked with your digestive system, with your gut, through your food, for example? Can it are you aware of things saying it can trigger depression, and how does it work?
I'm afraid we have to move on to the next opponent.
Okay, I'm sorry.
Thank you very much, Professor de Zuc. The next opponent is Professor Michetti. He is professor of neuroanatomy, and he is also a member of the National Research Council in Rome, and he is affiliated with the Università Cattolica del Sacro Cuore in Rome, Italy. Professor Michetti.
Thank you, Prorector. And thank you, Mr. Zhang, for your very interesting thesis. It's a very nice, interesting work, interesting presentation. I want to add my congratulations to you and to the proposers of your thesis. I'll do just some short questions owing to the restricted time. The first is a very general question, or maybe even better, a consideration. Your work is based on the experimental work, which is described in chapters 2, 3, 4, 5, and 6 of your very interesting thesis. At least at the moment when you submitted the thesis, as far as I can understand, just the work indicated in chapter 5 was published. Now I trust that all chapters have been published, and where or always which kind of problems did you find?
Okay, thanks your question, Professor. You want to know, now I just published in the chapter 5, and then another chapter I not published, or is right.
Mm-hmm. So there, but at least all of them are submitted since someone was just in preparation. Now you mean that at least all the others have been submitted, or some of them are in preparation?
Yes, and thanks. And first, the chapter 2, we will prepare has submit, and then another chapter we will have finished this and also modified with Professor, and we will publish. But and also in the other papers we also publish. We also do in my PhD studies, but just not at the matrix. Yes.
Yes. Okay. So you are refining the other papers, but I see in any case from your curriculum that you had many publications and your name is in a very good position. Okay. Another question deals with chapter 6. That is the effects of glyphosate on hippocampal plasticity. In that topic, which is a very interesting one, I have a curiosity. Maybe I've lost in in what you wrote, but I don't see any information concerning interneurons labeled by, for example, labeled by parvalbum. They are very interesting subneuronal neuronal subpopulation. Do you have any data, any information, although unpublished, on that topic?
Thanks for your questions. You might want to know the chapter 6, how is chapter 6 is right or not chapter 4?
I think it is chapter 6, the effects of glyphosate, yes.
Yeah, glyphosate, okay.
Yes, and herbicide Roundup, I think is the commercial name, yes.
Yes, it's maybe, yeah, it's not very focused on the immunotherapy. I know this, but this we can see. We find the 6 difference in the neuroplasticity. So it's connected with the chapter 5, and then I say the chapter 6 we find the neuroplasticity. Have the 6 difference, yes. So
yeah, that's chapter 5, yes. But my question was dealing with chapter 6, that is glyphosate and hippocampus. Maybe you did not test parvalbum labeled interneurons. It's just a curiosity if you had any information about that. No problem. Maybe you did not. Maybe you did not test this by immunohistochemistry. This some subpopulation of neurons. They are very interesting, in my opinion. That's why I asked you about this curiosity, but that's not very important. Maybe it's more important. Another question. The work is very interesting. I do not find out a strict connection with immunotherapy and depression. May you explain the connection of this work with immunotherapy and depression? Work is very interesting. It's quite a little bit difficult for me to find out the connection with the main topic of the thesis.
Thanks. So you want to know how the immunotherapy maybe connects the depression and the effects or something is right. And our study focus on is right. Yeah. Okay. So why I use the immunotherapy? The first is maybe first I want to introduce the immunotherapy. The immunotherapy is includes the just to enhance, regulate the body's immune response, and then to treat the disease. Then have some such as the biological or chemical or some physical and other different ways. But our study just focus on the biological immunotherapies because we target the immune cells and then the immune singulars. This is now the recent many people, many study are focused on here. But you know, actually, the immunotherapy is mostly used in some autoimmune disease, such as the cancer or tumor or some things. But here, and also I connect some other studies. I summarize some immunotherapies in the other paper that reviews that now immunotherapy is a good treatment for the depressions. Let's say, but
I have to interrupt here. We have to move on to the next opponent.
Okay.
Excuse me.
Okay.
Thank you very much, Professor Michetti.
Thank you.
The next opponent is Professor Drexhagen. Professor Drexhagen is Professor of Medical Immunology at Erasmus Medical Center in Rotterdam in the Netherlands. Professor Drexhagen.
Thank you very much, Pro-Rector, for this introduction. And certainly I also want to congratulate the candidate, of course, with his thesis. And also in China, Professor Cai Zhang, who I know for a very long time already. And we share, of course, our interest in the immunological background of major mood disorders. But to continue, dear candidate, perhaps you know or are aware that I'm the coordinator of a large EU project called Mood Stratification. And in this project, we already treat patients with thymostimulin as well as with low-dose IL-2. And we are now at the end of the period, and we start to evaluate our data. Perhaps you like to hear that we indeed find in patients also the very beneficial effects of low-dose IL-2, as well as be it a limited group of your preparation, alpha-thymosin. We also use the Chinese preparation, which we get from the Chinese fir. Now, I want to dwell a little bit on the low-dose IL-2. Although it's not in your thesis, I know in the group of Professor Cai Zhang, in particular, there is probably a colleague of you, Dr. Huang, who published on low-dose IL-2 treatment in also a mouse model of depression. Are you aware of that publication, candidate? Are you aware of this publication from your group on low-dose IL-2? So it's not in your thesis, but it's another form of immunotherapy, of course. Can you answer the question whether you are aware of that article?
Thank you, professors. I'm not very clear on this. You say some people have published about this about the immunotherapy in the depression or something?
Yes, on the low-dose IL-2 in a mouse model, and if I correctly pronounce it, of course, I do not exactly know, but the last author is Professor Cai Zhang, and it's from your same institute, Zhengqi Wang. Maybe Professor Cai Zhang can say.
Yeah.
So she or he, I don't know, of course, published on the low-dose IL-2. Are you aware of their data
yes.
or experiments?
Yes, thank you, Professor. And is interleukin-2, yes.
Yes.
They have published this low-dose, yes, interleukin-2. This paper is about the sleep induced the depression. So they use the interleukin-2 to treat, and have found it's a good how can improve the sleep and also can improve the depression behaviors. Yes,
correct.
It's a good.
Correct.
Yes.
So you're aware of that. So actually, in the patients which we treat with low-dose IL-2, there are two groups, two studies, one in Milano and one in Paris. They both give very beneficial outcomes for the depression, but the low-dose IL-2 has a very dramatic effect on the T regulatory cells. They increase immediately the T regulatory cells, and then we see a long-lasting effect of the low-dose interleukin-2. Of course, it is given in the groups which also receive a regular treatment as usual, so mostly serotonin or an antidepressant. So, but the effect is mainly via the T regulatory cells, and actually in this paper of Huang, she also, or he, or is he, I don't know, she shows the effect on T regulatory cells. Now I come back to your thymosin alpha-1. You know, we still have to evaluate the immunological changes induced by the thymosin in the patients. And I'm particularly interested in the T regulatory cells. You are focusing on the Th1 cells. Did you see anything happening in the you go for the Th1, Th2, but did you see anything happen in the Th17 T regulatory cell ratio as your group found for the low dose IL-2? Do you understand my question?
You say I use the Th1, the inhibitors, and how to there are some find about in the like the interleukin-2 or other cytokine exchanges.
What I want to know, in your model where you give your thymosin alpha-1, you see changes in your Th1, Th2 portioning, measuring gamma interferon and interleukin-4 in your serum. So it's a sort of correlate. But do you see also changes in the Th17 Tregs? Or didn't you see that? Because this is a thing which happens with low-dose IL-2, and it's strongly connected to the beneficial effects for depression.
Okay, thanks, professors. In our studies, because I use the Th1, so I just checked the balance about the Th1 and the Th2. I not checked the T17 and the T regular, this balance. Yeah, but and of course, I want to check, but because the Th17 and T regular, maybe in the chapter 3. I checked the triglycerides in thymus alpha-1 induced in the central or in the peripherals, but not the Th17. It's Th17 in the cells, in cells, not outside, not in the cells surface to check. So it's our machine can do this, but for the future, maybe I can, if we have a good flow cytometry, and then maybe we can do this or improve the experiment ways to do this or check sorry.
So, but did you check the cytokines related to it, like IL-17, TGF-beta, and IL-10 in your thymosin alpha experiments? Did you do that?
You may give a short answer because we have to move on to the next opponent. You may give a short answer, please.
Okay, I not do this balance. Yes, thank you.
Thanks very much for your answers. I give back the word to the prorector.
Thank you very much, Professor Drekshagen. The opposition will be continued by Professor De Baets. Professor De Baets is Professor of Neuroimmunology at Maastricht University. Professor De Baets, the floor is yours.
Thank you, Mr. Pro-Rector. Dear candidate, I would like to congratulate you also with your thesis and also include your promotion team. I have one question, a more conceptual question. Apparently from your thesis, you adhere to the pro-inflammatory hypothesis of depression. That means that depression is caused by inflammation or by certain cytokines. That's one side. You treat your depression, and we heard from Professor Drekshagen that's already used in humans also, with alpha-thymosin. And you say alpha-thymosin is a immune stimulatory molecule which is be used to treat like viral infections like hepatitis. So for me, it's a sort of contradiction. On one side, you say you have inflammation, stimulation of the immune system, and you're going to treat them with a drug that stimulates the immune system. I would rather give the patient, or in experimental models, a drug that inhibits this inflammation. So could you explain your thoughts about that? What I would like to propose, you first repeat my question in a short way so we can know that you understand it well, and then you can answer it.
Okay, thanks. You mean, or maybe you can just short to say this. Sorry.
So my question is, you have a sort of yin and yang effect. And the one side, you have pro-inflammation as the possible cause of depression. And the other hand, you're going to treat it with a molecule that stimulates the immune system. So for me, it's a sort of contradictory fact. So could you explain that to me, or try to explain that to me, why this contradiction? And apparently it proves to work, so but it doesn't fit to me with the pro-inflammatory hypothesis of depression.
In our study, we not to the inflammation. Sorry, it's we maybe you want to know our the immunomodulators we not to stimulate the inflammation. We just to regulate. Maybe we just regulate the immune cells, and then the immune cells then can release the cytokines, and then to regulate some neuroplasticity and all other things to induce the depression. Not the stimulants, it's just to regulate this.
So did I understand you well that you say your molecule, alpha-thymosin, does not stimulate the immune system, but sort of induces a sort of regulation. Is that what you're trying to say?
Yes, because the thymus alpha-1 is from the thymus. It's a nutrition effect. Because we also have a depression model, such as remove the thymus and then to induce the depression. So this just to promote the T lymphocytes mature and active the lymphocytes to release the cytokines to work.
But would it be more logical to use, I mean, we heard from Professor Dreksage that interleukin-2 was used, but would it be not more logical to use a cytokine like IL-10 to sort of restore the regulation or the dysregulation in depression?
Thanks, Professor. You mean the interleukin-2?
No, interleukin-10. Would that be more 10? Because, you know, that's a sort of suppressive cytokine.
Yes, because interleukin-10 generally is anti-inflammatory cytokines.
Yes.
So it's well to suppress the Th1 or something. Yeah.
Okay, I think, Mr. Prorector, I would like to finish this discussion and give the word back to you.
Thanks.
Thank you so much, Professor De Baat. The next opponent and last opponent is Professor De Rijk. He is Associate Professor of Neuroscience and Neuro-Urology, and he is affiliated with Maastricht University. Dr. De Rijk.
Dear Prorector, thank you very much. Dear candidate, I would like to congratulate you on this very elaborate thesis and this very international research team that has supervised you to achieve this wonderful result. I have a couple of questions about your work, and I would like to start by discussing your chronic unpredictable mild stress paradigm. And I looked in your papers, and I saw that the original article that you referenced for this paradigm was a study by Banasser in 2007, and that protocol was designed in male Sprague-Dawley rats, and they were only exposed to this paradigm for 15 days. And I would like to hear from you how you determined how to adjust your paradigm to 81 days in mice, and to in chapters 2 to 4, and to 49 days in Sprague-Dawley rats in chapter 5 for 49 days, if I'm correct.
Okay, thanks, Dr. Rejek. You might want to know why I use the different times between the mouse or the rats, right? The stress time is different.
Yeah, the different times between mice and rats, and the different times compared to your original source for the model. Okay, thanks.
About these questions, I want to say in my neighbors, we have done many studies in the mouse and the rats, and also compared the stress models in the these two mouse, and find the rat is easy to stress, but the mouse is difficult to create the models. So we use the different times, such as the rats, generally 6 or even 5 weeks, we can correct. But in the mouse, we cannot do this because we not check the depression behaviors in the mouse if we use the 6 weeks. Yes.
Yeah, that is clear, and that makes perfect sense. Yeah, I would suggest to elaborate on this also potentially in your articles, because you've changed the paradigm that you reference quite a lot if you use it for 4 or 5 times as long as in the original papers. Then I have another question. So as you mentioned, depression affects the lymphocytes at both the level of the central nervous system and the periphery. And we also know that depression not only affects the central nervous system, as you showed with your outcome measures of the sucrose preference test and the MACE test, but it also affects autonomic functioning, as you could assess by, for example, von Frey filaments to assess tactile hyperalgesia, or heart rate variability tests, or even continent behavior, so fecal and urinary continent behavior, to assess the effects on the autonomic nervous system. Is this something that you considered or would consider for future studies?
Thanks, but I can't get this idea. So can you just short to say this? Thank you.
So
you just no need to say this. Thank you.
So how would you, if you would incorporate this, how would you design a study to, for example, assess the effects of different pharmacological compounds on autonomic or central nervous system maladaptive changes due to depression?
Sorry, I also can't get this. Maybe you can ask a lot of questions.
So the compounds that you use in your study, are they specific for the central nervous system? Do they have effects on the periphery? And do you think that the effects that they have on the periphery could alleviate some complaints that are associated with inflammatory processes, for example, on the autonomic nervous system?
Or maybe you want to know the immunomodulators are used if they can pass the brain and then get into the brain, is right?
Well, no, I'm actually interested in their effects in the periphery. What do you think their effects are in the periphery?
In the because I use the IP to inject these drugs. So first they will work in the peripheral systems, and then they may be to go through the get into the brain, and then, because depression is the main cause the brain damages like the hippocampus or some other brain areas. So, but I'm not sure if these drugs can get into the brain, because this you need to check this. Yeah. But in our study, we not check this. Yes.
Okay.
But
Olga asked.
As you have understood, Mr. Cai Zhang, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis, and in particular the quality of your defense, and please await our return with the results of our deliberation. Mr. Cai Zhang, the degree committee here present online has assessed the quality of your thesis and also the quality in particular of your defense. And in view of this positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Steinbusch is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. I invite now your supervisor to take the floor. Professor Steinbusch.
Thank you very much, Pro-Rector. Dear Mr. Zhang, to start with, I would like to ask you the following. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful, honest, transparent, independent, and responsible in your science? What would you like to say to this?
Yes.
Okay, good. By the authority vested in us by law, and in conformity with the decision of the committee here present online, I hereby confer upon you, Mr. Cai Zhang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate, which will be signed by the rector, the secretary, who's here as well, but also both supervisors. We will sign this afternoon already for you, affixed by the official seal by the university, and now shown by our beadle. That will be the certificate which you're going to receive.
Thank you.
And I think we should make a little applause now for the candidate. And after this, I would like to give, several members of the committee of the promotion would like to address you, and I'd like to give first the words to Professor Chalier.
Well, thank you, Professor Steinbusch. Thank you, Pro-Rector. Thank you for all members of the Corona. And thank you very much for all people working with Cai. So just to give you a little bit of history that I have with Cai, he was supposed to join the lab actually a few years ago now, but COVID hit and it was hard to get Cai to come to France from China. But we did it. He came in 2022. He joined the lab thanks to Professor Steinbusch and Professor Zhang with two aims. He wanted to write, well, he had to write his thesis, and he wanted to learn a little bit more about other things. So he was forced by Professor Zhang, by Professor Steinbusch, and also thanks to Dr. Jody Belusky to sit at his desk and write his huge amount of data that he collected from China. He had everything, but nothing was written. But you did it, Cai, and I would like to congratulate you. But the second aim was also to learn a little bit about immunocytochemistry and brain anatomy. And so Cai was really eager to go back to the lab. Writing more than a couple hours was hard for him. So he had to go back to the lab. And when I say Cai had to go back to the lab, he physically ran to the lab. So Cai was running to the lab. He was running to my office, asking some questions, asking me to join him back to the lab. And he was running literally back to the lab. So this motivation, it's kind of amazing. And I do see also that the huge difference in his English when he came in the lab, it was extremely hard for him to communicate. And thanks to your will, Cai, and also thanks to other lab members, because I have to admit that Cai was surrounded by women in the lab. He has several master students, interns in lower levels, all women. And he was going mostly for like shopping, I know, once in a while, also teaching them what to do in the lab. So his communication skill improved significantly. So I know you did a whole lot of work, Cai, and I'm really proud of you. You did expand your knowledge about a whole lot of things. And I wish you all the best for the future. And I give you the, I think it's Dr. Poluski or Professor Zhang.
Yeah.
I think I'll go next briefly.
Yes.
Kai, first of all, I wanted to say congratulations on your thesis and the work that you've put into it. It's a job well done. And it was an absolute pleasure to work with you in Rennes. I do remember the first time that we were supposed to meet in the lab. I don't know if you remember. You were about a 15-minute walk away, but you ended up being an hour or so late. And you gave me a call because you had got lost. And if I remember correctly, Karitana, someone else in the lab, went out and found you and helped you come to the lab. But at that time, I remember thinking to myself, I hope this isn't an indication of how things are going to go in the lab. Because I'm like, if you can't locate the lab, how is this going to even work on a day-to-day basis? But in fact, I think being lost in translation quickly wore off, at least once you got into the lab and had your fixed routine. And it was very evident that you're very comfortable working in the lab. I also want to say I admire your courage to come to France, to leave China, to come alone to this foreign country, to join a lab in this country, and to work in a different language or languages, because of course, French is spoken in the lab as well. And so it's quite remarkable to do this. And I think the time you spent with us, I mean, I know it was invaluable to us and hopefully to you too. And I miss knowing that you were there to get things done and having these data sets sent to me regularly. So I definitely miss working with you. Your knowledge, perseverance, and dedication to research will be missed and is clearly evident, and I wish you all the best in your future endeavors. So I'll pass this now to Dr. Zhang.
Yep.
I'm deeply moved by this speech. Sorry, I have to adjust my speaker. Okay, sorry. I'm deeply moved by all the people comments and praise and to him, and it's my great day as well. Cai Zhang actually didn't have very strong background in China as a master's degree student, but he's my first student since I moved back to China from Canada. And he followed me, work hard through all these years. And then he finally went to Paris, France. And I think during this period he's in Europe, he not only learned from the science from you, and also learned see your high quality, seriously the spiritual to the science, and your kind and diligent personality to the student and to the colleagues. I think he learned a lot really from you, four of you from this period. And I can see he got matured and his English really getting much better, but these days he really go back to words. So I'm really worried. Go back to the original Chinese student kind of situation. I really don't want to take too much time. We're already delayed. I want to talk to Cai. This is the first step for your scientific career, and the long term to go and the long life to go to be a good scientist. Especially remember today all the questions, and you should answer those all the questions. Use your future time and to give the best answer in the future. Thanks to you all. And also I want to thanks to Cai for studying with me for so many years. Thanks everybody.
Thank you. So I will take the last part, and I was already informed that the total laudatio should not take more than 7 minutes. So we are over the time and I will keep it brief. So Kai, I would also like to address you. I met you for the first time in China itself. You came with Professor Song to a meeting which I had organized in Chengdu, in which we came with a delegation of Maastricht University to look to a possibility to have a collaboration between China and Maastricht University, and we did organize a big workshop. And at that workshop, Professor Song brought you with me and introduced me to you, saying, I have a student with me who might be interested to work with you. Then later, I visited you in Shangyang at the university. I spent some time. And one of the things I would like to mention to my other colleagues here in the audience is one thing I learned in Shangyang at the university, that everything is closing at 1 o'clock. And I mean, the entire university is going for half an hour or for 1 hour to take a power nap, but they call it over there. So everything was closed. The Mensa was closed, everything. We decided then we should like to bring you to Europe. You want to learn English, you want to learn immunocytochemistry, you like to learn more in neuroscience. And of course, we had the barrier with the language. And we noticed today, and I'm very frank about this, in your thesis, the language barrier was most evident this morning of you this afternoon. And of course, you left Rennes October last year, and that's now again almost 7 months, 8 months ago, and that's really a problem for you. I would really encourage you that you will be now an independent researcher. You are now Dr. Zhang, and you will go to international meetings, and at these international meetings, you have to speak English. So that's really, I would give it a priority. But for now, it's your day. It is your day in which you can shine. It's your day where you can see a next step in your career. And I really wish you a very good career in China. I think you already making steps over there. And I think there will be opportunities where I will be in China and maybe visit you, or where we have a possibility to meet you at our university. I also would like to take this opportunity, very brief, Pro-Rector, to also thank you guiding this officially, this ceremony. I'm really pleased with that you accepted my request. I also would like to thank all members of the committee over here, that you really took the time to spend almost 2 hours to sit here, and it's really a lot of time. And of course, I'd like to thank the entire promotion team, Professor Song, Professor Charlier, and Dr. Pawlowski, that they took the time to guide the students to bring them to this final level. Thank you very much. I give back the word to the Pro-Rector. Good.
Esteemed Dr. Zhang, dear Kai, it's my pleasure to congratulate you with your doctorate. And my congratulations are also on behalf of Maastricht University and of the Board of Deans. And I would like to share some impressions of the committee with you. We have enjoyed a very good presentation with a nice and clear summary of your thesis, very clear. And we also appreciated your thesis, good quality, an interesting work on an interesting topic and a relevant topic. And you have also used a variety of techniques so that you have supported your conclusions and your publications and your chapters with experimental evidence. Your defense was really for us difficult to judge. We are positive, but we are very aware of the fact that it was hampered by, I think, and we think, by nervosity. And also, we saw really struggles with language and understanding. And we take that into account, but we based our judgment mainly on the high quality of your thesis and of your presentation. But we are very happy that you are now a doctor in medicine. And I would like to congratulate your 4 supervisors with this result. And we hope also that you have already an impressive list of publications, that you will have also all your chapters and work published in the near future. So congratulations, Professor Steinbusch, Professor Charlier, Professor Song, and Dr. Pawlowski. We really congratulate you with the result that we have seen today. And I include, of course, Mr. Zhang, also your parents and other members of your family, and also all your colleagues and friends, and those people who have contributed to your studies and results as we have seen in your thesis. And I wish you a very successful professional career and happy personal life. Thank you, all members of both the assessment committee and of this degree committee. And in particular, I would like to thank all external members, and I would like to say that Maastricht University really appreciates your contributions in terms of a critical reading of the manuscript and giving your honest judgment and comments on both thesis and defense. Thank you very much for that. Before I close this ceremony, I would like to indicate that the committee will be online for another about 10 minutes to congratulate you in a confidential environment. And hereby I close this academic ceremony at Maastricht.